Hey guys, in this video, I'd like to go over a few things with you in Chapter 6. This is all about metabolism, and we're going to be starting to look at what energy is. We have a couple of other chapters dealing with some of the main energy type processes that take place in cells. We're also going to discuss a little bit about enzymes, and so as you go through this chapter, make sure you are finding some way to learn vocabulary. I know I've mentioned that before, but this particular chapter really does have a lot of very good vocab words that you're going to see here, but also in some of the upcoming chapters as well. And so the outline just kind of goes over a few of the main sections that we'll look at. We're going to look at cells and how energy is kind of goes through a particular type of flow, or kind of changes form there. We're talking about different types of energy. Specific types of metabolic reactions and pathways, enzymes as part of this chapter as well. And then we'll end briefly talking about what an oxidation-reduction type reaction is. So let's just get started with section 1, cells and the flow of energy. Again, these are some basic energy-related terms. Many of these you have probably seen before if you've had a previous science class, biology class, even like physical science. Or chemistry is going to talk about these terms as well. But energy, your basic book type definition for energy, is the ability to do work. And so if you think about it, there's a lot of different types of energy. It could be, and of course we're going to have some other definitions here. Kinetic energy, these are a couple of categories of energy to think about. But think about the energy that you see on a day-to-day basis. Like the sunlight provides energy. Well, that's different than the energy that's inside of the food that we eat. That's different than the energy that's inside of the cells, our cells needing ATP energy. We'll kind of look at that. That's different than cells. That's different than energy that you see as far as electrical type energy. So there are different types of energy. Two main categories, and again, you've probably seen these terms before: kinetic versus potential. Anytime you think of some type of kinetic energy, it means that energy is in motion. It's a moving type of energy. It's doing some kind of work there. They give you kind of a little basic example. If you can imagine a water going over a waterfall, there is energy there. That's why we have hydroelectric power plants, because they try to utilize that type of motion energy, that kinetic type energy, and that's a type of mechanical energy that falls into that category. Potential energy, sometimes you hear this called the energy of position. It's whenever something is able to store up energy, and it's like it's ready to be used, it's ready to be converted into kinetic energy, something like the food that we eat. Now, our food that we eat, you can think of it as a fuel source, really. Things like carbohydrates, that provides energy for us. Fats, those provide a lot of energy, and especially long-term type energy. And so that food that we eat, we're going to use some of that energy right away, but we also store a lot of that energy. And so that's a potential type of energy when you store it and kind of have it ready. But then you need to be able to access some of that stored energy as well, and you have to convert it into some kind of kinetic energy. So there's different types of energy. We're going to look at some of those as we go through as well. If you've had some kind of physical science class or even basic biology in the past, you may have talked about the laws of thermodynamics. We're going to look at these here: the law of conservation of energy and the law of entropy. The first law here, the law of conservation of energy. Energy cannot be created nor destroyed, but instead it changes forms, right? So when we're talking about the energy that our body needs, we take in food energy, and then we need to use that food energy to get the energy our cells need, ATP. We're not creating the ATP, we're just transforming, we're changing the chemical energy that's in the food, carbohydrates and lipids, all these sugars and fats and lipids and things. We're taking that type of energy, we're changing the form and turning it into something that our cells can actually use. So it's a conversion. We didn't create new energy. We're not destroying energy. Energy cannot be created or destroyed. the chapter kind of gives an example of photosynthesis. And so we'll have a whole chapter dealing with photosynthesis. You can kind of imagine, you know anything about plants and photosynthesis, they have pigments there that kind of capture or trap that solar light energy. And then the plants use that energy to form carbohydrates. They take some raw materials like carbon dioxide and water, they chemically combine that, and they form a chemical energy out of that solar energy and those other raw materials. Second law, thermodynamics here, the law of entropy. Entropy says when energy goes through this transformation, when you do change the form of energy, there is always a little bit of energy that gets lost, right? So, or there is some kind of amount of energy that doesn't get used for that work that you were trying to accomplish. In other words, no energy conversion is 100% efficient. You get some loss of energy. Does that conflict with the first law that we just said? It's not that energy was destroyed, but it has not really been utilized to its fullest capacity. Up at photosynthesis, even something like photosynthesis is not 100% efficient. Our cells, when our cells go through doing this chemical change, we're talking about taking food energy, turning it into ATP. That's not 100% efficient. Not 100% of our chemical energy and carbohydrates and lipids can be converted directly into ATP energy. I like to think about how your car works, right? If you're familiar with a combustion type engine, you put gasoline in it, but how much energy do you really get? How much work energy can you get out of that gallon of gas? You think about how much heat. If you've ever looked under the hood and you feel all that heat that engine has generated, there's a lot of essentially wasted, in some sense, loss of energy just because of the heat that's lost. Right? So those are laws of thermodynamics. Some illustrations there that kind of gives an idea. You have the solar energy here for photosynthesis, put in some raw materials, carbon dioxide and water. You get the glucose that you're trying to make, the carbohydrate that the plant is trying to make, but you also lose some energy in the form of heat. Same thing about animal cells. Here shows a couple of muscle cells. Muscle cells are going to use up some carbohydrates in order to go through a type of muscle contraction. But even that's not 100% efficient. It's going to create some heat. Now, if you're thinking about animals and thinking about humans, we do give off a lot of heat, but our bodies need some of this heat. So maybe it's not really lost and never really utilized. Our body temperature, you probably realize, is somewhere around 98.6 degrees Fahrenheit or 37 degrees Celsius, and that's pretty warm. And so we need, some of this heat to be used there that you're generating. But in other words, the conversion here is not 100% efficient. And so the processes that are taking place inside cells, they involve all these different types of energy transformations. And so what that means is there's not as much energy that's directly available to do some kind of work that you intended to, but that's okay. Organisms that are called producers, we're going to look at this later in another chapter, but producers use that energy to create organized structures in biological molecules. In other words, plants that are producers. They use solar energy, they go through photosynthesis, they store the energy into biological molecules such as carbohydrates. And then consumers consume producers, they eat plants, they eat that energy, use this potential type of energy, the stored up energy in the carbohydrates, transfer that or convert that, transform that into from potential to some kind of kinetic energy to drive their own cellular processes. All right, take a look at all of these vocabulary words in section number 2 on metabolic reactions. We have metabolism. Metabolism we've probably heard used before. You think of energy usually when you think of your metabolism. How is your metabolism? Are you got a lot of energy? Are you able to burn a lot of calories? That's your metabolism. Um, really, in a biological sense, really you're talking about any kind of chemical reaction taking place in a cell. That's really what your metabolism is uh, in a biological or a chemical type understanding. And that involves things like reactants and products. Now, these are chemical terms, chemistry terms. So if you've had some chemistry and you've looked at a type of a chemical formula, you know that what goes into a reaction, those are your reactants. So if you think about a chemical formula on the left-hand side, you've got the reactants going in, they react, and then you have products that form as a result of that type of chemical reaction. You're going to have these things in a metabolic or metabolism type reaction. Some metabolic reactions build up and form more complex compounds. So in other words, you could take little small subunits. Remember when we talked about like taking an amino acid, combining that with other amino acids, and you form proteins. That's a building up type of a reaction. It is still a metabolic type reaction. Sometimes you're doing the reverse. You're taking a larger macromolecule, maybe like a complex carbohydrate, or even like a disaccharide, a sugar that has two subunits there, and you're breaking it down into a simpler form. That's still a metabolic chemical reaction. Sometimes you see the term free energy, how much energy is available to actually complete this type of reaction. Some of these reactions are going to need to, you're going to have to add energy to that reaction to get it to actually work. Some of them don't. Some reactions will happen spontaneously. They don't require an added form of energy. And so those definitions are here for you as well. Exergonic reactions, the products have less free energy than the reactants, right? So in other words, they're releasing energy. They're actually producing energy. So the products, those things on the right side of the equation, what you're producing, they have less free energy than the reactant. So that's actually an exergonic reaction is going to give off some kind of energy. It could be you mix a couple chemicals together and it starts getting warm. Anybody ever use like the little warm packs, instant warm packs? You kind of break something inside the pack that mixes the two chemicals together and it forms heat. Well, that's a type of energy that reaction is giving off. Or if you know about the little glow sticks and you kind of break the little vial inside and you kind of mix the two chemicals and now you're giving off energy, but it's in the form of light. It gives off energy. Exergonic gives off energy. Endergonic reactions, the products, the book definition, the products at the end of the reaction have more free energy than the reactants do. In other words, there's a requirement to put energy into this. These are not spontaneous reactions. You could mix these two chemicals together and they wouldn't do anything until you added some form of energy. You add energy to that reaction and then that would drive that chemical reaction forward. When we talk about energy for cells, we've been kind of mentioned chemical energy that the body needs, carbohydrates and lipids, and technically your cells can get some energy out of proteins, although that's not the first choice, I guess, for cells is to kind of get some glucose, sugar type energy, and then maybe some stored up carbohydrate. In animal cells, it will be glycogen. In plant cells, that will be starch, or start breaking down some fats, lipids. But really, your cells are going to have to convert that, transform that type of chemical energy into ATP. You've never seen this before. This is adenosine triphosphate. It is essentially nucleic acid. All right, it has a base. It has 3 phosphate groups attached to it. All right, high energy compound that's used to drive different types of metabolic reactions inside the cell, building up proteins, getting your muscles to contract, having the cell division type pathways we're going to get to eventually, any kind of cellular energy demand that the cell needs to have, it's usually going to be requiring that energy in this form of ATP. Now, generally, cells don't store up this particular energy, although there is a kind of a cycle that takes place. I like to think of ATP as like a fully charged, if you think about rechargeable batteries, everybody's got something that has rechargeable batteries. You got your cell phone, you're going to have to recharge it at some point. ATP is like when you charge up your cell phone battery and it's at 100%, it's good to go. You can use that energy to power your cell phone or to power whatever electronic device you're using, whatever it is. But then once you use up some of that energy, you have this molecule left over called adenosine diphosphate. This is less energy, right? This is less energy. Do you just throw the battery away? My cell phone dies, right? I don't just get rid of it. I have to charge it back up. Well, the same thing happens in your cells. This ADP is formed. It's a lower energy molecule. You don't just get rid of it. You want to recycle it, really. You want to add energy back to it so that you can use it again in the form of ATP. All right, there's where I was trying to get to at the beginning. ATP, it has the adenine, that's the base. It does have ribose. That's your 5-carbon sugar, and so that's called adenosine. And that has 3 phosphate groups. We know it's 3 because it has the tri right there, triphosphate. And so this energy generally works by coupling with some type of a energy needed type of a reaction. And so the energy released by an exergonic reaction, exergon is going to give off energy, is somehow kind of captured by this ATP molecule. And then that ATP molecule is used to drive some kind of reaction that requires energy, and that's inorganic reaction. Here's your little quick figure 6.3 that kind of shows you the adenine base would be here, the 5-carbon sugar would be here. You got 3 phosphate groups here. It's really the energy in this bond of the phosphate group that really is that potential energy storage area. And so whenever you break this bond, that releases some energy that can be used to drive a biochemical type reaction inside the cell. Once you break that bond, you have ADP. I'm not sure if let's see, I think we go through some of the figures here. Once you break loose that phosphate group, and that's kind of what this is showing you, you can kind of break loose that phosphate, and now you've released some energy into the cell. That energy can now be used to drive forward some kind of inorganic reaction, some type of reaction that requires energy, and it forms ADP. The ADP is diphosphate. It only has 2 phosphate units left, and that's what this picture was kind of down here. ADP, the only difference here is that it only has 2 phosphates instead of 3 attached to it. Remember, this has lower potential energy. It doesn't mean it doesn't have any energy. This has lower potential energy than the ATP does. Again, you don't want to get rid of this molecule. You want to save it so that you can recharge it, add energy back to it. To do that, you've got to add another phosphate back to it, because remember, this is diphosphate. If you add a phosphate ion back to it, then that strengthens that molecule, and that now you've restored the potential energy of ATP. All right, section 3 talks a little bit about some metabolic type pathways and enzymes. Usually think of reactions as taking place in a particular sequence of events. You start out with your reactants, and then you have products left over. Sometimes you've got some kind of intermediate. In other words, you don't always have, a reactant automatically going to the product right away. Sometimes you've got some little intermediate forms. It may have a series of steps that it has to progress. And as we're going to see in the section, many times it requires some type of enzyme to change the amount of energy requirement of that particular reaction. The other thing is, it says products of an early reaction sometimes become reactants of a later reaction. We're going to see that really used as a word called a substrate, and we'll see that when we talk about enzymes briefly. And so the little diagram here is kind of saying, okay, in this little series of steps or kind of a chain reaction type pathway, I start out with my initial reactant. Usually the initial reactant, the first thing you start with, could be called a substrate. Those words are usually used interchangeable. That substrate, anytime you're talking about an enzyme-based reaction, usually you're going to see this called a substrate. Right? It's basically your reaction, your reactant going, what's going in, the chemical going into the reaction, the reactant, it would be your substrate when you're dealing with an enzyme type reaction. That's usually going to produce maybe an intermediate somewhere. Doesn't necessarily mean it's going to have 1, 2, 3, 4, 5, but it may have some type of intermediate type molecule that's a kind of a go-between before you get to the end result. Now, they don't really change the name of the end result. They just call that a product, just like in a basic chemical formula. So the really only difference here is that you've got a reactant, a product. Sometimes the reactants are referred to as substrates, and then you may have one or more intermediate molecules, compounds formed before you get to the actual product. Now, we said all that, kind of look at enzymes. You may already know a little bit about enzymes. First of all, these are usually proteins. Probably 95, 98%, really maybe even 99% of enzymes fall into this category called proteins. Occasionally there are like an RNA type molecule that serves as like an enzyme. But for our purposes, most of the time we think of enzymes as being proteins. They function as catalysts. That's a good chemical vocabulary word, chemistry term. It just means that it helps to speed up a reaction. It catalyzes a reaction. It speeds up a reaction. We're talking about an enzymatic pathway. Again, the reactants, instead of calling them reactants, they change the name. They're going to be called substrates. They speed up their reaction. I've already mentioned that. They accelerate a specific reaction. We'll see some examples of that. Each reaction in a metabolic type chemical reaction requires a unique and a specific enzyme. Not all enzymes are the same. Not all enzymes are going to work with the substrate. In other words, the substrate or the reactant and the enzyme have to match. They have to be specific. Now, all enzymes do have, or most enzymes do have some similar characteristics, and we're going to talk about those. If there are intermediates in that chemical pathway, this end product, in this case, G was our ending product, will not result unless all of your steps are complete. In other words, if this is a more complex type pathway, you've got your substrate here is A, you got your product there is G. This has 5 different intermediate steps. If one of those steps was missing in this particular pathway, you wouldn't form your product. It would just stop. Right? If you're missing an enzyme, if you're missing enzyme 3, and this is just an example, if you're missing enzyme 3, you'll never go from this intermediate C to this intermediate D. Everything would just stop at that point. You could go from B to C, that's no problem because you would have enzyme number 2. But if you're missing something along the way, your pathway stops. All right, so there's something called the enzyme-substrate complex. Turns out that enzymes, and I'll show you the diagram of the figure that goes along with this in just a second, but it turns out that an enzyme is going to have what's called the active site. The active site is where the substrate actually binds to on the enzyme. Remember, substrate in this sense is really the reactant that's going into the chemical reaction. And so the active site is where that reactant, that substrate binds to the enzyme, and that creates what's called a complex. Once it binds there, it actually causes a conformational change. It's just a very slight alteration, but it causes that enzyme to change shape at the active site. Once that active site kind of changes shape, it's almost like it pushes those molecules together, those reactants a little closer together, and it allows the chemical bond formation to happen a little easier. And that's what helps to speed up these enzymatic type chemical reactions. You may have heard of the lock and key concept, and that's kind of the lock and key hypothesis. That was kind of the way this was described at one point, and that still applies because that suggests that you have a specific enzyme that reacts with a specific substrate, and that still applies. It's like how a lock will only accept a specific type of key. In other words, my key that operates my vehicle only works with my car, and if I try to unlock the door in your car, my key's not going to fit that. And so the lock and key hypothesis still describes that type of relationship of enzyme and the substrate specificity. But they also have what's called the induced fit model, and that tries to describe what's happening at that active site. So once the enzyme and the substrate bind together, it kind of causes a change in again, a conformational change in that protein in the active site, and it's like the enzyme is induced to fit or induced to accept that substrate, and you have like the optimal now conditions to try to get these two reactants, if you have two reactants, to actually come together and go through a chemical reaction a little easier. When we look at the kind of a diagram, this is from the chapter textbook there. This kind of helps us illustrate. This is from another figure we're going to be looking at in more detail in just a minute, but it kind of gives us a couple of terms here. First of all, we see the enzyme. The enzyme, usually a protein, it has some type of location on it where the reactant will bind to. That's called the active site. The active site is where your substrate or reactant molecule or molecules, could be two, but in this case it's showing one molecule here binding to the active site. Notice that it's kind of cut out in the diagram here to represent that the substrate does have to be specific, a specific fit to that enzyme, and that forms what's called that enzyme-substrate complex. What would happen next is once it binds, you would have that induced fit. In other words, you would have a little change in the shape of the protein enzyme here, and that would kind of perhaps bring these molecules closer together or somehow allow this chemical reaction to take place a little bit easier. Generally think of two main types of reactions when you think of enzymatic reactions. The first one is degradation, which means you're breaking down something. You've got a single substrate, single molecule that's going to be a reactant. It binds to the active site. It forms the enzyme-substrate complex. It goes through the it kind of changes shape a little bit, and then your substrate is released into the products. Okay, so degradation, you start out with a single molecule, and you come out with more than one product there. Notice here, though, I'll kind of outline one of the main things about enzymes that we learned kind of at the beginning. One characteristic of enzymes is that they're not used up in the reaction. They're not part of the products. So that's why this diagram here shows the enzyme substrate complex. It shows the products being released, and it shows the enzyme kind of being restored back to its original shape, its original conformation. So in other words, that enzyme can be used again. You can take that enzyme and you can reuse it. Theoretically, you can continue to reuse it over and over again, unless something happens to that active site that causes it not to be usable any longer. A synthesis type reaction, that's just the opposite. You're actually putting two molecules together. So you begin with two substrate molecules, two reactants are going together, and they still will bind. Even though you have two separate molecules, they'll still bind to an active site that's specific to those reactants. They still form that enzyme substrate complex. You still have enzyme that is kind of not really left over, but is now ready to be used again. And in this case, you have a single product that forms. So that's a synthesis type reaction. What's happening here really, and one thing to kind of think about, enzymes is that they do speed up reactions, they do serve as catalysts, but one way they do this is by lowering the amount of what's called the activation energy, the energy of activation. Most chemicals, most molecules have to have some kind of energy input. We kind of talked about endergonic, exergonic reactions. Most reactions have to have some kind of energy input in order to activate them and in order to go through that chemical reaction. And so enzymes help in this process by lowering how much energy is required in that particular reaction. So they lower that energy of activation by bringing those substrate molecules in closer contact with each other. And that, of course, takes place on that active site of that enzyme. And so one thing that influences is the rate of the reaction. How fast is that reaction going to take place? Well, that's influenced by a couple of characteristics, and we'll kind of get to that. Take a look at figure 6.6, and it kind of illustrates what would happen with just a regular chemical reaction without an enzyme and then with an enzyme. And so here on the x-axis, you see the progress of a chemical reaction. There's kind of like a time feature here. And then the amount of energy required, or free energy, is there on the y-axis. And so whatever kind of reaction you're looking at, it kind of starts out, it requires a certain amount of energy here. If you look at the activation energy without the enzyme first, that's the line in blue, enzyme not present, then it requires a certain amount of energy. They didn't really measure that. They just said, okay, take a look at the bracket. You have to get this so much amount of energy in order to get this reactant to be converted into these products. But if you have an enzyme present, a specific enzyme that these reactants can bind to form a complex, notice, it drastically reduces the amount of that energy. So now it's a much easier and much quicker, using less energy to get this product to form. That's kind of the idea behind the enzymatic reactions, trying to lower or helping to lower that amount of energy to get that reaction going to begin with. So we'll talk about a few factors that influence this rate of reaction. And most of these are pretty easy to kind of think about. The substrate concentration. In other words, the more starting reactants that you have, the more concentrated your substrate will be, usually increases the enzyme rate, the enzyme activity. Now, there's a limit to how much that that will take effect. If we go back and look at the little diagram here, you can kind of imagine if you have more substrates here, then that allows the rate at which you're converting substrates into products to speed up, as long as you have enough enzymes there to bind to those substrates. Like if you don't have enough enzyme present, then it doesn't matter how many more substrates you add. You're not going to speed up that reaction if you don't have enough active sites to bind to. But provided you have plenty of enzyme active sites, then by having more substrates, you can actually produce more products. Optimal pH. This is very important, especially for living things, living organisms. And so enzymes have usually a range or a specific pH level that they work best at. And so usually it's at a neutral or a slightly basic pH or slightly alkaline pH. And this figure here is kind of showing you an example of two enzymes, pepsin and trypsin. They both function to break down protein. Pepsin is actually found in the human body in the stomach. If you think about the stomach acid that you have, it's a very acidic concentration of substances, acids, and this pepsin works very, very well at a low pH. In fact, its optimal pH is around a 2, which is very close to what hydrochloric acid would be. And so this enzyme works very well at a very low pH. It can digest proteins best at a low pH. Whereas trypsin, this is not found in the stomach because it just doesn't work at a low pH. It's actually found in your intestines, in your small intestines, where it can help to digest proteins once you neutralize that stomach acid. But But it just shows you that, some enzymes work better at a low pH. Some enzymes seem to work better at a closer to neutral, or maybe even slightly alkaline or basic pH here. Temperature, that greatly affects pH. Usually enzyme activity will increase when the temperature increases, and that has to do with just molecular motion. You increase the temperature of molecules, they speed up a little bit, they move a little bit faster, and so the chances of them coming in contact with the enzyme and the active site, all that kind of increases, and so you generally increase the reaction rate and you increase temperature, as long as you don't go too high. So enzymes, again, have a specific optimal temperature, just like pH. They have an optimal temperature. If you get too hot, it can actually break down the protein structure. Remember, like the 4 levels of protein structure, just that linear primary structure and the secondary structure, the tertiary and the quaternary structure of proteins. If it gets too hot, that structure of protein will denature. It changes the actual structure of the protein, and it just won't function. Really, the same thing happens in pH as well. Having the incorrect pH, less than optimal pH, can also start to denature proteins as well. There are something called cofactors and coenzymes that we'll look at. Cells can regulate the presence and absence of an enzyme. These molecules help to do these 3 things here. They can regulate the concentration of the enzyme. They can activate or deactivate some of the enzymes. These are molecules. That prefix co means together, and so an enzyme cofactor is a molecule. Usually it's like an ion a lot of times, that functions to, or a mineral that functions to bind to the enzyme, to either activate it or deactivate it, or to kind of control its function a little bit. So it works together with an enzyme. So most enzymes don't just work alone. They have to have a cofactor present. A lot of times you'll see that magnesium ions are used as a cofactor. Magnesium, when it binds to the enzyme, kind of gets that enzyme started up, ready to go, activated. So here's a couple other examples. Molecules, cofactors, molecules required to activate enzyme. We'll talk more about these. You don't have to memorize these. FAD, NAD. These are cofactors that we'll talk about when we talk about photosynthesis type reactions. But they're molecules that are required to get the photosynthetic enzymes activated. The word coenzyme refers to an organic molecule. These are organic, so they have carbon, hydrogen, oxygen. They're not protein-based, and so a lot of times living organisms require what we call a vitamin. Just like in our diet, we have vitamins. Most of the time, vitamins are functioning, not always, but many times, vitamins are functioning as a coenzyme, and that helps to also kind of regulate the function of an enzyme, control a little bit of what an enzyme is doing. So they're working together with an enzyme. Another topic associated with regulation is what's called enzyme inhibition. You got to have an ability to control, regulate the function of enzymes. And so there's 2 examples here we'll look at: competitive and non-competitive competitive inhibition. Sorry about that. Anything that inhibits the enzyme activity can be an inhibitor. Right? Inhibitor, as the name implies, is something that slows down or helps to regulate the enzyme activity. Sometimes that molecule works by competing with the active site. And so this is that same diagram we looked at before on degradation, but here's a substrate molecule. Again, the reactant, the enzyme, the active site forms a complex. But if you can imagine, I didn't draw in I should have drawn in like another molecule, but if you can imagine another molecule here that had a similar shape that would actually bind to the active site, that would be a competitive inhibitor. It's some type of molecule that that's able to bind to this active site but doesn't cause that, it's going to block essentially that substrate from binding. You can imagine some kind of molecule that kind of fills up that space where the active site is, then your reactant, your original substrate, wouldn't be able to bind there. So that's competitive inhibition. It's like this molecule, this inhibitor, is trying to compete for that space, compete for that active site. Non-competitive inhibition, your chapter actually has a picture of that. I'll show you that in just a second. Non-competitive inhibition is kind of is similar. You still have an inhibitor, but instead of binding to the active site itself, there's some other site on the enzyme that this molecule binds to. And so it's called an allosteric site. Don't let that word confuse you. Allo means other. So if you see that prefix, allo, it means other. So there's some other site on the enzyme. What happens essentially is if that allosteric type inhibitor, that molecule binds to the enzyme, somehow it causes that enzyme again to change shape at the active site. So now that active site no longer binds to that original substrate. Because again, it's a lock and key type scenario. If you change the active site, you change the shape of the active site, then the substrate can't bind there. That's the non-competitive inhibition. That's the allosteric type scenario. If you block the active site with another molecule, that's your competitive inhibition. Right? So those are good vocab words. Kind of make a flashcard with those, or draw out a little diagram or picture. Kind of keep those straight there. This figure 611 is kind of trying to show some type of enzymatic pathway, and you've got some kind of enzyme here, and then you've got some type of allosteric. Here's your enzyme here. Sorry, here's your enzyme here, and you got an allosteric site, the other site that something could bind to. And once it binds, it kind of changes the shape now. And so now my active site no longer, here's the original substrate. Now my active site no longer will fit the, my original substrate. So I can't form, the product that you're trying to form, that sort of thing. Last section introduces us to oxidation-reduction reactions. We're going to see a lot of this again as we get into some of the energy-based reactions, photosynthesis, cell respiration, metabolism. But just to kind of introduce some things. If you had some basic chemistry, you probably talked about redox reactions, oxidation-reduction type reactions. The main couple of things, main takeaways here is right here. The oxidation is loss of an electron. Right? If you lose an electron, that molecule becomes oxidized. Reduction is gaining an electron. And so essentially, redox reactions, you're going to be kind of passing electrons from one molecule to another. And we're going to see that when we talk about the processes of cell respiration, the processes of photosynthesis. And so I was kind of looking at this little example down here in the production of sodium chloride. Sodium is oxidized and chlorine is reduced. Maybe if you had chemistry, you may have thought about this little mnemonic device. Try to memorize that. Oil rig. You've seen that. Oxidation is loss or losing electrons. Rig stands for reduction is gaining. That's kind of how you can kind of remember which one is which. The other thing I kind of like to point out is remember the electrons represent energy. And so if a molecule gains electrons, it's essentially gaining energy. And we'll talk about this, especially when we get to photosynthesis type reactions. But and also cell respiration, if you reduce a molecule, it's gaining electron, it gains energy. If you oxidize a molecule, it loses electrons, it essentially is losing some energy associated with that molecule.